Hello judges, my name is Jordan Hurley and today I will be presenting on the deactivation of bacteria from silver nanoparticles. First I want to discuss how silver nanoparticles are a possible alternative for antibiotics. Through the widespread use of antibiotics, this has resulted in many drug-resistant bacterial strains, which in return has called for a need to eradicate them. Silver itself has been used throughout history for various medical purposes, including as a disinfectant, for wound management, as well as a blood purifier. In the early 2000s, researchers began to notice the antimicrobial potential of silver nanoparticles and how they could have a significant value to the medical field. They have the ability to destroy multiple drug-resistant strains that antibiotics alone cannot. What makes them also appealing as an alternative is that they are non-toxic, very cost-effective, and their synthesis is a fast and green alternative. Although the actual mechanism for how these silver nanoparticles can cause cell death in many different bacterial strains is unknown, there has been a few different mechanisms that have been accepted widely throughout the science community. This includes the infiltration of the cell wall of targets as well as protein deactivation of targets from binding silver ions. So in our work, we will be utilizing these silver nanoparticles as a photosynthesizer in photodynamic therapy due to their antimicrobial potential, which I discussed earlier in the previous slide. What photodynamic therapy is, it's a medical procedure to deactivate bacteria by utilizing a photosynthesizing agent and light irradiation. In our work, we also utilize methylene blue as a photosynthesizer due to traits that it possesses that I will discuss later in the presentation. So in photodynamic therapy, an injection of the photosynthesizer is made into or near a region of the epidermis which has experienced trauma. This region will then be stimulated by a light of a specific wavelength, and this causes the production of singlet oxygen from the photosynthesizer. Singlet oxygen is known to be toxic to cancer cells as well as bacteria, so any bacteria that may be present in that region will be eradicated using this method. So the way that we synthesized our silver nanoparticles in our work was through liquid phase pulse laser ablation. And we used various different mediums to synthesize them using this method, including citrate, PVP, and DI water. But it's really important to note here that we needed to make sure that there was a narrow size distribution of the nanoparticles in order for them to be an effective photosynthesizer. The images above that I provided captured using an electron microscope or the TEM show the average diameters of the nanoparticles that we synthesized as well as their standard deviations. So in our research, our bacteria was cultured in LB broth. At 37 degrees, it was incubated for a 24-hour period. Our bacteria that we cultured included E. coli as well as Staph aureus. And after the 24 incubation period, there was a concentration of 10 to the 6 colony-forming units per milliliter, which is what we utilized throughout our research. Agar plates were then made and inoculated with both types of the bacteria, and each bacteria samples were then combined with silver nanoparticles alone at different size distributions of 10, 20, and 40 nanometers, methylene blue alone, and a combination of the silver nanoparticles at each size distribution as well as the methylene blue. The diagram on the left shows the combination of these bacteria samples for reference. After these plates were made, they underwent light irradiation for 5 minutes utilizing a 660 nanometer light, which falls in the visible spectrum to be red light, as you can see above. So according to our results, the methylene blue and silver nanoparticle combination was the most effective at eradicating both the gram-negative E. coli and gram-positive Staph aureus after 5 minutes of light irradiation. With these conditions, we were able to eliminate all of the bacteria present. Methylene blue is able to generate copious amounts of singlet oxygen, which can react with the outer membrane of bacteria and further damage it. This makes it an excellent photosynthesizer, which is why we utilize it in combination with the silver nanoparticles and also explains why it was the most effective at eradicating the different bacteria cultures of our samples. It's also important to note that as the size of the nanoparticles decreased, there was an increase in the antimicrobial potential. And this may be due to the fact that as you decrease the size of the nanoparticles, their surface area to volume ratio decreases, so they have a stronger ability to attack onto the cell membrane of different bacterial cells. And it can also be observed that the size of the nanoparticles as well as the methylene blue had the most significant impact on the antimicrobial effect. You can also see that the silver nanoparticles alone were most effective at eradicating the Staph aureus than they were the E. coli, and this may be due to the differences in their concentration of peptidoglycan. So we utilize ABMDMA, which is a singlet oxygen collector, to determine how much singlet oxygen was being produced from each one of the samples as through light irradiation. So we were able to see that the light generated more singlet oxygen whenever methylene blue was present. And whenever more singlet oxygen is present, this actually decreases the optical density or the absorbance, of each sample. So as you can see from the diagram, whenever methylene blue is present, as time went on through light irradiation, the absorbance continued to decrease. This suggests that more singlet oxygen is being produced with a combination of methylene blue, which explains why the combination of methylene blue as well as the silver nanoparticles was the most effective at eradicating the bacteria. The experiment was also conducted again to demonstrate the effect of the mediums that the silver nanoparticles had on its antimicrobial effect. After 3 minutes of light irradiation, we found that the DI water was the most effective compared to PVP as well as the citrate solutions. And this may be due to the fact that the silver ions are attracted to hydroxyl groups, which are present in water. And so this results in the release of more silver ions to attack the cell membrane of the bacteria. So in conclusion, the decreased size of the silver nanoparticles resulted in the increase of their antimicrobial potential on our bacteria samples. We also found that when synthesized in DI water as the medium, the silver nanoparticles were the most effective at eradicating the bacterial samples. Our research further proves the effectiveness of silver nanoparticles as a photosensitizer for photodynamic therapy. I just wanted to thank you all for taking the time to listen to my presentation, and if you have any questions, please feel free to contact me or Dr. Ern.